Thanks for the invite here and today's lecture on how to interpret genomic variants in general and how network knowledge of the cell may be useful in doing that is quite close to everyone's hearts. So let's see. As all of us know or should know in this audience, massive DNA sequencing is underway for lots of different diseases. We happen to mostly study cancer, in which case you have, depending on how you tally it up, about 10 to the 5th genomes. Of course, that depends if you're talking about the complete genomic coding sequence or just the exome portion of that. But nonetheless, it's a lot of genomes, and it's made possible by these X10 instruments, among other advances. And if you read essentially any of those cancer genome atlas papers that people have been publishing, or again, for any complex disease, by definition, practically, the most surprising finding, or one of the most striking findings out of all of those studies, is how enormously heterogeneous each patient's genome is. So in cancer, when you sequence the genome, as again you may know, you have actually two genomes. You have the genome of the tumor, and then you have the genome of the normal tissue, and you subtract the two to get just the mutations in the tumor relative to the normal. But even then, you have on average, depending on the mutational load and tissue type and all of that, let's say about 100 mutated genes in that tumor versus normal. And so if you look at that pattern of somatic mutations, it's essentially a snowflake for that particular patient that has never before been seen in nature and will never be seen again in nature, that 100 gene set. So, and that really is the fundamental challenge that underlies interpretation of these complex disease genomes. To see that in a couple of different ways, let's first just randomly take a patient out of the Cancer Genome Atlas. Most frequently mutated gene among the 25 genes this patient has mutated is GATA3, and that's about 50 times more than expected by chance in terms of the background mutation rate. This is relative to background mutation rate. And then very quickly you get into this long tail, this quote unquote long tail of mutations. And then this line here is itself the random background. So notice there are genes that are mutated below random backgrounds. Now, are these genes functional or are they not? And so notice GATA3 is a known cancer gene. BRCA1 is perhaps the most infamous breast cancer gene, is not currently detectable by GWAS, but it just gets missed. But of course, it's known by lots of other means. And then these two genes are also known cancer genes, yet they're very rarely mutated in this cohort, begging the question is, are they functional or are they not? So that's the real key challenge of heterogeneity. Let's now switch cohorts and look at the Cancer Genome Atlas ovarian cancer. And now we're going to zoom in just one chromosome, gene by gene resolution, not mutation by mutation resolution, but gene by gene. So there's about 550 genes encoded on chromosome 17, and a dot indicates that that gene was mutated in that patient. For a long time in cancer biology and in complex disease biology as a whole, that this disease is not a disease at the nucleotide mutation level or even at the gene level, but it's a disease at the pathway level. Here's the whole pathway. And although one gene in that pathway in particular, in this case EGFR, is frequently mutated, here in 57% of these glioblastoma patients, look at how many more mutations you can pick up and recognize really is the same subtype of cancer based on the fact that genes in that same pathway or downstream of EGFR here are mutated. And look, you have genes like RAS, which are very well-known cancer genes, only mutated in 1% of this particular patient cohort or population, but nonetheless, we believe these are going to be functional because they're hitting that pathway. And again, it's not just RAS, but it's really many different points connected by pathway interactions. So that's the working hypothesis for how we might make sense of all of these data. The question then is, how do you implement that hypothesis in any kind of systematic way? And that's right now where the field is. We sort of know that pathway information is important, but how exactly you get at that in the same kind of systematic rigor or with the same kind of rigor that the Genome Project itself is getting at mutations is sort of the open challenge. And my last introductory slide here is just to remind us that most cancer genes have not been discovered with sequencing or with GWAS. So if you take, and this is again part of Matan's paper that's in press right now, he did a very interesting thing, simple to do but no one had done. If you take the gold standard set of cancer disease genes, and you might say, well, which one do I believe your gold standard set? There's about 3 different ones he took. This one is the cancer gene census from the Sanger Center. It's got about 500 cancer genes. They're all literature curated and validated by that team. But if you prefer a more conservative list, you might look at Bert Vogelstein's list, and there's other lists you might look at. All of them are going to have the same qualitative plot that I'm going to show you here. For each accepted cancer gene in this database, what Matan did is went into PubMed, and you might know PubMed has these things called mesh terms that actually I've never personally used before, but were really useful here. So if you looked at the first PubMed publication of each of those cancer genes that associates that gene with cancer. So not the first time the gene's ever mentioned in the literature, but the first time that someone says this is a cancer gene. And then you look at that PubMed mesh term annotation, it tells you what were the experimental methods that were used in that paper. And he records what those experimental methods were. And this took us through 2013 here, although he did this actually in 2015, but I think the database, he wanted to make sure he had a completely updated cancer gene census, so it ends in 2013. But you can see here of those genes, the real winner in terms of the first discovery of that as a cancer gene is gene manipulation by knockout, knockdown, knock-in. So the original Bob Weinbergian experiments where they showed oncogenes by transfecting in a DNA fragment that would transform cells. That was certainly mapping to the gene knock-in or gene manipulation category. Another great example to pick out of here, P53, although it is the most recurrently mutated gene in sequencing and would certainly be rediscovered by sequencing, was in fact found first by gene expression. So it would map to RNA analysis here, in fact. So you could always say, well, sequencing has come along more recently. Maybe part of this is historic, and that could be true. But it's nonetheless, I think, sobering to look at this and realize that sequencing really has not found most of the cancer genes we now know, and it's worth finding out whether it could. Okay. Oh. So then the question is, how to get at these cancer genes and how to get at the pathways that these cancer genes function in any kind of systematic way. For that reason, late last year, Nevin Krogan and I announced this initiative we call the Cancer Cell Map Initiative. It's just getting off the ground. It's a collaboration currently between UCSD's Cancer Center and UCSF and their Cancer Center and the 2 teams there. But the idea is twofold. One, can we integrate knowledge from genotype into these networks as a way of predicting and better predicting phenotypes of interest to clinicians like disease diagnosis, response to therapy, and so on. And essentially, the operation is exactly the same as like all good geneticists we do to go from genotype to phenotype, the difference being that now we want to insert a middle layer, and that middle layer would be a comprehensive map of pathways relevant to that disease biology. Begging the second question, how do we begin to accrue that comprehensive network knowledge in any kind of sort of systematic way? Much like genomes are now systematically sequenced, can we do the same for pathways? Today I'm going to talk almost exclusively about this first task of, I'm going to assume we have a lot of network knowledge and we're going to get a lot more of it as systematic pathway mapping continues, but the question really is the analysis challenge that I'm going to address today in my talk for how you would use that knowledge systematically to translate genotype into clinical phenotype. And for that purpose, because we don't yet have incredibly systematic knowledge of networks and pathways, it's coming via a bunch of different technologies I won't spend much time on today, we do at least have existing network knowledge, and that comes and has been collated in a bunch of different databases that many of you, I think, in this audience probably, if you don't use, you've at least heard of, alphabet soup of acronyms like BioGrid, INTACT, Human Protein Reference Database, and so on and so forth. These collate interactions across many different kinds, protein-protein interactions, transcriptional or protein-DNA interactions, metabolic connections. You can always throw up your hands and draw a co-expression network if you want to do that to connect together co-expressed genes. If you sort of just back of the envelope look at how many interactions you can get from existing databases, it's about 1 million. It's about 10 to the 6th for human pairs of human proteins. This, of course, is by no means comprehensive. It by no means has a well-characterized error rate. These databases are not context-specific in terms of what disease or cell line, or much less patient, that they refer to. But nonetheless, you get a large collection of, again, about 1 million interactions that may or may not be useful as hypotheses behind the pathways of interest. So that's what we're going to do here, just to get started with this whole exercise. And so for about 10 or 15 years, my lab has worked on ways of translating patient data through networks in this way. But the paper I'm going to talk about today is this 2013 Nature Methods paper, because it was built on the shoulders of a lot of our earlier work, for instance, done by Han-Yu Chang and Alice Lee. This is Matan here. But I think this paper had all of the wisdom of those earlier ones, and it really has been pivotal also for all the thinking in my lab since. And so the way that this method uses networks to interpret patient data goes something like this. So if you simply look at the raw somatic mutation data, as we already saw in the ovarian cancer example, you cannot cluster it. In fact, what's the first thing you want to do with omics data? Not knowing anything better to do, let's try to stratify those patients by their omics data. That is to say, cluster the patients by their omics data. For gene expression, you can always get clusters of the gene expression profiles because it's not a sparse data set. Every gene has a level in every patient, and there's lots of downstream effects like proliferation and stress that cause patients to be quite similar to one another across the whole cohort. And so you can always get clusters. The question then, are they meaningful? In the case of somatic mutations, we saw you can't get clusters. And in fact, if you simply try to cluster patients right out of the box, you get one monolithic cluster in this particular variant of the clustering algorithm, or each patient in their own cluster. It just doesn't know what to do because, again, there are very few features shared in common between patients. Everyone has a p53 mutation, and after that, no one has anything in common whatsoever. That's sort of what's happening here. So here's what Matan did. He said, well, under the hypothesis that it's not the gene level that's causing these patients to look similar or not. It's the pathway level that we want to analyze. So what I want to do is transform those gene measurements by pathway knowledge into something that I can cluster. And so essentially, I'll show you a sort of schematic of the approach in a second. But what this approach is going to do is, instead of representing whether a gene is mutated or not in a patient, instead of just using that list of genes, every gene is now going to get scored in a patient by how close it is to mutations in the network. So even if a gene wasn't mutated in the patient, if that gene is upstream or downstream of the mutated gene, it will feel heat and it'll get a score. And so now you've transformed a very sparse set of 50 to 100 genes into a network proximity score for every gene, and that clusters beautifully. Let's see that. And so first, the algorithm here is not original. It's got several different names depending on which literature you read, but they're all similar: network propagation, network diffusion, or network smoothing. The paper I like to refer people to that was sort of an early use of this in computational biology literature is this paper by Vanunu et al. From Roded Sharon's group at Tel Aviv University, published in PLOS Comp Bio in 2010. The first use in cancer I'm aware of is by Ben Raphael's group in this paper here, and we're using a very similar algorithm now to cluster patients. The way it works is, for each patient, you propagate those mutations on the network in a way I'll describe. So imagine two patients, a yellow patient genotype and a blue patient genotype. Yellow marks the mutated genes in patient 1, blue marks the mutated genes in patient 2. Maybe there's one gene like P53 that's mutated in both patients. And now it runs essentially a thermodynamic simulation where every mutation is a source of heat, and that heat can then spread over time through wires in the network, which are these protein interactions. So then that blue heat now spreads to its neighbors, and then to a lesser degree to the neighbors' neighbors, and so on, until the entire system comes to equilibrium. That's essentially how it works. There's a closed form solution for the math, but that's not important. The point is you get this sort of heat spreading, and that's how upstream activators, repressors, and downstream effectors will feel the heat if you've mutated here. So in this toy example, what's happening is because this region of the network gets hit a lot in both patients, even though it's almost never the same place, the whole part of that network now feels heat in both patients. And that similarity is what allows you to cluster the patients in a robust way. So to show an example, this is now from the ovarian, back to those 351 ovarian cancer patients. This happens to be the network responsible for clustering the patients robustly into one of the 4 clusters. In fact, 2 slides ago, that slide I showed that took an unclusterable data set into 4 clusters. This is now one of those 4 clusters. So the idea here is, although 2 patients in this cluster almost never hit the same genes, every patient in that cluster hits one gene at least on this subnetwork on this slide. Just to kind of illustrate one case study there, this cluster of red genes happens to be the fibroblast growth factor receptor and fibroblast growth factor family. So it's getting connected in the network database, presumably here, because you have a family of related genes all in the FGFR family. A few of these genes were already known to be cancer genes, FGFR1 in particular, because it is frequently mutated in cohorts of cancer patients. It is hit time and time again over that cohort. But in other cases, you have a bunch of N of 1. So there's one patient that hits there, for instance, one patient that hits there, one patient that hits there. If you didn't have network knowledge, even though this is almost, in retrospect, an obvious case to have looked at this gene family, it wouldn't have popped out. You would not have found any recurrent events. Now, what the network is doing is organizing them all at a higher level into this network. And I wouldn't show you this particular example if it weren't also clinically interesting. So once you get clusters, whether it's mutations and networks in this case, or it's gene expression, or whatever way you've derived your patient clusters, you'd like to know whether those clusters are clinically informative. Now, up until this 2013 paper, there had been about 8 different expression studies of ovarian cancer. And while all of them had shown a clustering figure at some point in the study, none had been able to link those clusters to any kind of clinical outcomes. And so we thought it was significant that when you cluster with mutations as opposed to expression, you got clinically informative outcomes. And so here are the survival and the drug response curves for those 351 patients, stratified by the network subtype. So the network I just showed you, when it gets hit, that's the blue subtype. It's the most aggressive subtype. You can see after 70 months of time, no patients are left alive. Compare that to the best subtype, where 65 upwards of that % of the patients are still alive. And if you look at the drug response in terms of platinum sensitivity, by the way, all ovarian cancer patients get the same standard of care, platinum-based agents. Why? Because we can't stratify them right now. So they all got platinum, and you can therefore look at what fraction remains sensitive to platinum over time, and you can see that what's essentially happening here in this aggressive subtype is all these patients are drug resistant, essentially right out of the box. Now, this is unpublished data that we're writing up now. So since the 2013 study, a second large sequencing study of ovarian tumors came out by the sister project of the Cancer Genome Atlas, the ICGC, or International Cancer Genomics Consortium. We were quite pleasantly surprised to see that when those patients came out, even though it's fewer patients and one of the subtypes drops out for lack of patience. If we now simply use these subtypes in a prognostic way, or so in a supervised way, for you bioinformaticians out there, then we can take each one of these ICGC women and put her tumor into a subtype that we defined back here, and look at the survival of those cohorts, and it's the same exact stratification. So that's suggestive. If you look at the back again at that network I showed you, this is a quick and dirty layout of the exact same network I showed in the previous slides, same nodes and edges. If you now look at those nodes and edges, at which ones are getting hit in TCGA in orange, which ones are getting hit in ICGC in blue, the green are the genes that are hit in both cohorts, you can see that without the network, you would see much less similarity, just 4 genes here, than you can now recognize between these 2 different continents of patient cohorts with the network. And so that's kind of, again, showing you how the network is able to integrate these disparate events. And then just to sort of give my one non-cancer example, my lab mainly thinks about cancer, but we collaborate with many others who think about all sorts of complex diseases. So this is a paper by Joe Gleason from a couple of years ago, where we used essentially the same approach that Matan had developed. And this is sort of the network that integrates mutations for that disease. This is more of a straight gene linkage / GWAS type of paper for a rare childhood neurodegenerative disorder called HSP, or hereditary spastic paraplegia. Joe sequences these cohort of consanguineous individuals as a way of mapping disease genes for these rare childhood diseases. And essentially what he was able to do here is use that network to substantially increase his power to find linkage for some of these candidate genes. And it's the same exact idea. You heard from Pamela already, we developed Cytoscape. I have to have my obligatory advertisement for Cytoscape. But I also want to point out that our latest thing is we are trying to get Cytoscape hooked into a back-end database of networks. And there's lots of interaction databases out there, but what we saw a need for in the field is databases of pathway diagrams that come out of studies like are going on here in many of your labs, like go on in my own lab and many labs everywhere. You find that you have a pathway diagram coming out of these systematic network analyses, or quote unquote subnetwork. Maybe one of those subnetworks ended up in a major figure of your paper, but then 7 others are like deep in the supplement. And you might know them, but no one else is ever going to know them. The idea is we need to capture those. These are not canonical pathways like you'd find in textbooks. There's databases for those. These are working databases of network hypotheses that have been published, so they're somewhat mature, but they're not all the way to mole bio of the cell textbook kind of thing. And so you can, like Facebook, you can define social groups. You can share your networks at the beginning with just your work group. Once you publish, you can make those networks public or not. It's up to you. Hopefully, you would make it public once you publish. This was actually developed by initially a grant from pharma. And so, for instance, Roche has index deployed inside of Roche, and they get all the public networks, but they also get just the Roche-specific networks that none of us can see. An interesting feature, but it's useful for industry collaborations with academia and so on and so forth. So check that out. But now for the remainder of my talk, I'm a little more than halfway through here, I want to talk about where we think we should go from here, and which is essentially the same thing as saying what bothered us about that last analysis. And what bothers us about that last analysis, and in fact what bothers us about network biology as a field as a whole, is that networks do not look like the contents of cells. The contents of cells are not networks. It's not the same as if you opened up a Pentium chip. There is in a Pentium chip a network that is the wiring diagram of transistors and connections between those there. And so this hairball we draw for biology actually does exist in some domains when you draw that hairball, like in electronic engineering. In biology, you know, okay, two proteins interact, we draw a line between them, but that's not what the cell looks like. The cell looks like this. And in fact, for effect, what you're looking at here is the same structure in two different representations. This is the proteasome. As you may know, the proteasome is the vacuum cleaner of proteins in the cell. It sucks up and degrades proteins. You're looking at that vacuum cleaner from a side view. So there's multiple folds of symmetry, of course, but you're looking at the side of that vacuum cleaner. This is in orange and yellow tones. That's the core of that vacuum cleaner, and then the regulatory particle is in purple and blue on either side. And that same color, so there's the core, it's here, and in purple and blue, there's the regulatory particle there. You can already start to see there is some similarity between the network representation and the structure, because in the spring-embedded layout in Cytoscape, you can see it's trying to pull apart the core and the regulatory particle, because there's a higher density of interactions on either side than there is between them. The question we had is, how much farther can you push that towards ultimately its structure, or if not a structure, at least some sort of hierarchy of modules that structure contains. And so let me describe what I mean by that point. So the proteasome, it'd be great to get a 3D picture of it, but even before you get a 3D picture, you can at least recognize that there's like a deep 3-layer hierarchy of modularity, even in this quote-unquote single protein complex. There's the whole proteasome. That factors into a core and a regulatory particle. Each of those, in turn, factors into parts, and it turns out there's even a sub-factoring beneath that you can't see here. So what we tried to do starting a couple years ago was see how far we could get at that hierarchy of modules that a protein complex or pathway contains directly from data, from interaction data, I should say. And so that resulted in an approach we called NEXO, for network extracted ontologies. So why the word ontology? Because if you think about it, the gene ontology is in fact such a hierarchical factorization of the cell. So I'm expecting 90% of my audience probably has used the gene ontology and either loves or hates the gene ontology, or maybe both at the same time. But if you haven't, the gene ontology, the easiest way to describe it is it's the best model of a cell, at least descriptive model of a cell, we as biologists have, for better or for worse. It's a large team of about 5 different groups headed by Judy Blake that has attempted to factor a cell into its constituent parts and their parts and their parts and their parts, all the way down to individual genes. So at the top of the hierarchy, you have organelles, and then organelles factor into processes, and processes factor into complexes and smaller complexes, and finally genes. And we thought, and this is now a team led by Mike Kramer and Janusz Dukowski, that we could try to reconstruct a lot of this hierarchy directly from network data. We went for this process first into yeast. So the next number of slides are no longer human cancer. It's now all in Saccharomyces cerevisiae, because we have by far the most network data, or really any omics data, for yeast as for any biological system or species. And if they pour in different interaction types, like protein-protein interactions, other kinds of interactions, and start to cluster those interactions using any of about 1,000 different clustering algorithms that have been published, you could begin to create these binary trees or dendrograms to describe your clustering. You would join 2 genes based on their similarity in interaction space, and then you would join that later based on similarity to other clusters, and so on and so forth, until you had this entire tree linking all genes. That starts to look like the hierarchy of processes in a cell, with 2 key differences. One is, when you're talking about, say, a protein complex in the cell, there's no requirement that that complex only has 2 genes beneath it, or 2 proteins. A protein complex can have how many proteins at once? So we first need a clustering algorithm that can join right away. If you have 7 proteins or 7 objects that are equally similar, it should just join them in one fell swoop. That's the first requirement that most clustering algorithms didn't have. And the second is we have this notion of pleiotropy that geneticists call pleiotropy. That is to say that what about a component, a gene or a protein complex that works not just in one parent process, but in multiple parent processes? Why not just recognize that in data? Why force everything into one cluster? Let the data tell you how many modules that gene or complex participates in. So you need to allow for multiple parents, and then you get a structure that is driven not by some computational convenience like this binary tree that really came out of evolutionary biology, right, and speciation trees. Here, we need a structure from our clustering that resembles and is driven by data. And so that's all I'm going to say about the algorithm. It's published here in Nature Biotech in 2013. You can read all about it. We updated that in bioinformatics in 2014. And you can read all about that. In fact, I think there's probably a lot of work to still be done in how you best do this clustering, but we've got a couple of tools. And having built this structure, you can now directly align it against the reference human or literature curated version that we call the gene ontology. So you can recognize when you've rediscovered, there's the ribosome, I just rediscovered that. There's something else, there's protein translation as a parent of the ribosome. I just discovered that. And then you can also look where you have not captured the Go and you have novel concepts or terms that you have found there. And that's your novel biology. To bring this a bit more down to earth, this is an unpublished project. This is Mike Kramer's last work before he graduates my lab, where he's trying to build build a entirely data-driven ontology of the process of autophagy. As you may know, autophagy, like the proteasome, degrades proteins and components in the cell. It's the other pathway by which you can recycle components in the cell through the so-called lysosome or vacuole. In the yeast, it's a vacuole. But just to show you how it works in this case with data, so we now here, we've already integrated lots of different omics data sets, again, PPI, protein-protein interaction, gene expression, et cetera, into an overall pairwise gene similarity score. So red means in data these genes are more similar, blue means they're less similar. So and then we've already done a 2D normal clustering on a subset of core autophagy genes here to show you how it works. And so you can, as many of us are used to doing, look at these heat maps and pull out by eye some of the structures. So you can see here ATG7 and 3. ATG, by the way, stands for autophagy, if you didn't guess. ATG7 and 3 form a tight cluster. But look, 7 forms another cluster with 10 and 12, and 10 itself forms another cluster with 27. So already you can see a straight clustering would have not really done the right thing here. And not only that, but then all of these, so here's a case where that strong cluster now has a slightly less strong cluster above it and a slightly less strong cluster above it still. So all of that, by the algorithm, gets processed and you get this ontology. And by aligning that against the knowledge in the reference gene ontology, you can now start to name the terms or the concepts or the clusters you have found in this heat map. So let's look at what happened with ATG7. It got put up to 2 terms. And then all of these guys end up getting clustered here, which looks a lot like what Go calls core machinery of autophagy. Now, you get orphan terms that don't align to Go, and so it turns out one of them is actually here, 183. That's just the systematic numerical name that the algorithm gave it. You look at this term, though, in the data, 1027. That's a pretty strong cluster, and it turns out it's very robust. If you look at the data, there's multiple experimental data types putting those two together. That's probably a real function, and it's very clear where to look. That's the idea. Let's now look at the whole ontology for yeast. So not just one sub-process. This is what it looks like. In fact, you can browse around it. There's an online tool here that let me just quickly show you after I orient you. So the root of this thing is here. All the names, again, come from just direct transfer from the gene ontology database by direct alignment. Very quickly, so this is the whole cell. The cell factors into a mitochondria and a membrane and an intracellular part. This is, of course, again, all directly from data. Why did it do that? Because genes that are mitochondrial genes have weak similarity to one another. It's nonetheless stronger than to non-mitochondrial genes, and that's why you get that whole branch. And it turns out that Go tells you that was the mitochondrial part. Let's now look, and just to bring this whole thing full circle, let's now look back at the original example that motivated me here with the proteasome. There's what the proteasome looks like if you currently look at the network in Cytoscape or your favorite network browser. Here's what it now looks like in this Nexo data-driven hierarchy or gene ontology. That's up towards the root. So just to go back, here it is in the full view. And just zooming in on that, and again, the names come from transfer from Go when you recognize the same term. So we found the proteasome. It splits into a core and a regulatory particle. The core, in turn, splits into an alpha and beta subunit. The regulatory particle splits into a base and a lid, and then you have all those proteins. So you have this 3 layers of hierarchy that do get recapitulated. And just so you'll check out this, there is a tool behind this. So here it is online in nexontology.org. This is more of I think a publicity stunt right now than to actually have a functional tool, but you can see how useful it might be. So now we can zoom into this thing. There's the proteasome, right? And if you want to actually see what are the raw data that led to any one of these terms being formed, you can click on it, and it's going to pop out, and there's the hairball, okay? And if you want to look at the interactions, maybe it's a little more intuitive here. You can scroll down and get all of the data that are supporting that particular inference. But again, why do that? Just look at the hierarchy and realize that data have gone. All these data have gone into building it. Oh yeah. All right. So in the last 7 minutes here, one more thing to talk about. So just to finish off that point, maybe when we're looking at these hairballs, we're simply too close to the data. Not unlike if you think again about the structures we're going for and their functions. How is that structure actually generated from structural data? Well, it turns out that's a cartoon that I downloaded from PDB. But if you click and you find out where the data actually came from, it's a few million X-ray diffraction images stacked together and analyzed by now a very well-vetted set of algorithms that we call structural proteomics. Right now, you take all of those images, you push the button, and you get that. Not that simply, as you know, but that's the workflow. Of course, you can look at those pictures, and people do and certainly did, but that's obviously much more intuitive. So in the same way, maybe networks are just like diffraction images. It's not something we can process by eye easily, but there's important patterns there that can be analyzed if you know how. Okay, now I want to get back to the first thing I talked about, which was using this kind of information to interpret genomes. So I showed you at the beginning of my talk how through the network you can use that in a flat way to integrate mutations that can stratify patients and predict things like survival and drug response. Can we do the same thing now that we've changed the representation of networks and we're no longer looking at them directly, but we're building them into hierarchies? That's what this last piece is about. Again, it's still in yeast because our hierarchies are in yeast, but you can see where we would like for this to go with more research. And this just came out a few weeks ago. So the way this is going to work now, so first I have to have a model genotype phenotype translation problem in yeast. I certainly have them in abundance in human disease. Here, our model problem was predicting yeast growth phenotype from simple genetic perturbations to the genotype. So in yeast, lots and lots of experiments exist where a single gene or a pair of genes or 3 genes, a triplet of genes, have been deleted and the growth rate measured. The largest such data set is by Toronto, by Brenda Andrews and Charlie Boone, who looked at many double gene deletion genotypes and scored them for growth. And combining all of the extant data out there in the literature, you get what I think is probably the largest genotype-phenotype compendium in existence. It's about 3 million genotype-phenotype pairs. Again, most of those are pairs of genes being deleted and a growth. So it's a huge amount of genotype-phenotype data in yeast. And the question is can we predict the growth for a simple genotype like a pairwise gene knockout through this hierarchical network representation or through the gene ontology? And an ontology is a very natural way to think about integrating these data, because it spans perfectly, or in a very convenient representation, all of the scales between genotype and phenotype. If you think about this, it really is, genotype-phenotype is the original multi-scale problem. You have insults or changes to nucleotides at the level of nanometers, right? Those impact protein complexes and larger molecular machine at the order of hundreds of nanometers. Those then impact signaling pathways, and finally organelles, and finally the cell. And the cell's at about, for yeast, 1 to 10 microns. For humans, maybe a little bit bigger. So we basically connected here a 1 nanometer scale to the cell level, which is maybe a 10 micron scale, and all of the layers in between. If you talk about how deep is this hierarchy, Go is about 12 layers deep. Our data-driven Go, Nexo, is also about 12 layers deep. So it's quite nice as a translation hierarchy or device to get from a nanometer scale to a micron scale. The way we thought about this was take the genotype, which is by definition, a genotype is the set of states on genes, and convert that to a set of states on intermediate biological processes like complexes, pathways, and organelles. We call this the ontotype. So whereas the genotype is sets of states, there's a state of genes, the ontotype is the set of states of all biological objects, all the way from genotype to phenotype. So for instance, this large complex T5 here would get some state which is a function of its components and their components all the way down to the genes. That begs the question, what should the function be that integrates all of this? And that's an open question. Here we wanted just to start as simply as possible, so we used an OR gate. Essentially, if anything below T5 gets mutated, I call T5 mutated. And in fact, as a slight variant of that, what Mike Yu has done here is simply count the number of genes beneath that term that's mutated. And so like -2 means that there's 2 genes that have been deleted beneath T5 here when I delete B and D together. And notice that's the same ontotype I can get from deleting another set of genes together because they're impacting the same hierarchy of processes. Once I have the ontotype, now I just do a straight GWAS or machine learning thing, and just in a black box way, try to associate these term states with the growth phenotype. That's the way it works. I'm going to skip that slide and go right to the prediction. So here, interaction score is really just applies to double mutants. You can think about this as the predicted growth score versus the measured growth score. This is, again, against those 3 million genotype-phenotype relations in yeast. It's in cross-validation if you're interested in that. And you can summarize how well the predictions are working. I mean, certainly you can see here some correlation, but you can actually, of course, compute a correlation or a precision recall from the raw analysis. But let me focus really on this set of bars, because what it does is it just takes this analysis and gives you a correlation. So what you're looking at here is about 0.34. It's a correlation coefficient between predicted growth and measured growth. So room for improvement, it is what it is. And this is using the Go hierarchy. This one is using the Nexo data-driven hierarchy I described. So you can see the performance whether you construct the hierarchy or you take the literature curated hierarchy. It's about the same. At the moment, the literature curated one wins slightly. If I randomize the structure of the hierarchy, I, as a negative control, like to see that my correlation goes away. And then there's a bunch of papers that in the past have attempted to predict these kinds of growth for genetic interactions using what we'll just call non-hierarchical approaches. There's a bunch of different ways, and we just chose 3 of them here, FBA, MNMC, and GBA. And for comparison, we also simply flattened both the GO and the Nexo networks. So you might be aware of there's straightforward ways of taking the entire hierarchy and just reducing it to gene similarities, and just using that gene similarity network for prediction of interactions. And essentially, all of these non-hierarchical approaches perform around the same and significantly worse, I hope you'll appreciate, to the hierarchy of, or these hierarchical approaches. And again, here's the precision recall versions of that same analysis. So we think there's something simple and inherent to this hierarchy that's really working for us here. So that's my talk. Where do I think this is going? We want to apply this to human cancer and other diseases, but generally we think it's a really nice way to proceed here to move from genotype to phenotype. Why? Because it's a way of capturing cell biology. Networks just capture cell biology. These hierarchies now just encode those networks in a way that relates genotype to phenotype. Because genotype to phenotype, I used to think about them as fundamentally qualitatively different objects. We have genotype, we have phenotype. They're not the same. They are the same. They're just 2 different scales. They're traits of biology at 2 different scales. And those 2 traits on nucleotides and traits on cells or on organisms, can be connected through hierarchies. And so you can think about mutations hitting genes or dripping down from genes through this sort of organic system of funnels, and then ultimately all getting coalesced to create a unified phenotype that we see. So in summary, as we know, genome sequencing has revealed thousands of genes altered in many diseases. Some of these genes are well known and have strong effects. Others have weak effects, and we don't know exactly how many there are. But common patterns emerge or begin to emerge at the level of components, pathways, and systems. For cancer, Nevin and I have recently launched this open campaign to try to map cancer networks and apply them using these principles. And this is, by the way, not meant to be an exclusive activity. We'd love to involve people who are interested in and have a planning meeting going on into the summer for that. So please let me know if you'd like to participate. And then in this last piece, which I think it is certainly the most interesting computationally and maybe the most forward-looking biologically, that we think network data are just too close to the raw data, and we should be building at least hierarchies from them, if not something even more advanced like a whole cell model. And we have some promising results that show how that representation is even better at connecting genotype to phenotype. And it's this idea of ontotype, that really those 2 notions, genotype and phenotype, are just opposite ends of a spectrum. So with that, I will take any questions. Thank you very much.
Thank you for a fabulous talk, and there's usually lots of lively questions afterwards, and I won't even have to ask the first one because there's already one. So.
Thank you. So I guess I have like 3 very related questions. So you mentioned the Go biology, ontology, and in terms of how you were able to kind of capture some of the biology of this, right? Have you looked at other kind of like cell pathways, if you will, and try to see if maybe some of these mutations cluster in certain specific groups of pathways, like in the Hallmarks Cancer paper that kind of described multiple processes that have to be hit in order to...
I mean, that's exactly where we want to... so the idea is now we'll have a hierarchical representation of that, or we need to work towards that. And again, I think it's going to be interesting to see what that hierarchy is. In terms of the hallmarks, it's basically a 2-level.
Mm-hmm.
Is that right? Maybe I should go back and read that paper, but you know, at first blush, it's basically 1 level of hierarchy. You have these 12, I think it is now, pathways, and
Sure.
they have genes. But we know it's going to be like 12 layers deep.
So, I mean, like, have you tried to see if, like, let's say, the cake pathway, just using that to perhaps group together your genes, see if that works as well?
Yeah,
as
so
your gene
again,
expression
I
based?
think these part of that's been done by the TCGA. I mean, you
Mhm.
know, like that glioblastoma pathway I showed at the beginning.
Mhm.
Certainly, my group is not the first by any means to organize mutations in canonical pathways. And KEGG, I think, is one of the sources people use, especially for instance, glioblastoma, where you have IDH, which is a prominent TCA cycle gene that's frequently mutated, and so you have sort of a rich story around metabolic canonical pathways you can integrate. So I think in my talk, it's really been how would you systematize that?
Mhm.
And clearly at the end, I think you should go back and ask how much of the systematic discoveries overlap with hallmarks.
And one last question is, I'm interested in complex diseases, particularly Alzheimer's. But what are your thoughts on, like, the application of this to kind of discover epistasis and, like, epistatic interactions?
Yeah, exactly. So epistasis is an interaction between 2 or more genes, just to be clear, right? And that's exactly what has gone on here to get this good predictive performance over yeast growth. So essentially, all these experiments where Toronto knocked out pairs of genes and measured growth, that was to find epistasis. And so you're really trying to predict that the game here really is. If you just want to predict growth, 90% of the time you can do pretty well because you can start by predicting wild type growth, or at least just the single, you know, you can look at the single gene deletion phenotypes and just add them together. If you have 2 or 3 gene perturbations, you would just add the effects of each perturbation independently. And so you can predict growth fairly well that way. But here the question is, what can you do to predict the actual interaction? So that's, if you notice the predictions versus, let's go back here, too fast. I glossed over it. This actually wasn't predicting growth directly. That would look like 99. That would be perfect correlation, and it wouldn't be helpful. But here we're actually doing the harder problem of predicting the interaction. This is predicting epistasis, and so that was actually the benchmark we used. I sort of glossed over it. This is all about how well you're predicting epistasis.
Hi, Trey. Hi, this is Ray. I'm a PI. I work with Eric. I was at the UCSD, so probably we met in Uri Hall doing my postdoc. So basically, I have 2 questions. One is relating to the first part, that you use this mutation data to stratify the patients. Have you find the concordance when you use the mutation data to stratify the patients with the patterning that you use the gene expression?
Great question. So I mentioned only gene expression to sort of diss gene expression and say, oh, there's 8 papers that tried this with gene expression and they failed, right? That was, but so let's now get back to that. So we've now discovered subtypes. Those were not discovered before. So we've defined a novel stratification of this disease. That stratification appears to be clinically useful. Why wasn't it found before? And that's question 1. And question 2 is, having found it, can you train expression profiles to find it? That is to say, can you select a few genes whose expression levels do give you those subtypes? And the answer to this second question is yes, you can. So I can go back. Let's start with that. I can go back and, in a supervised mode, find mRNA levels that do predict those clusters. So now, why weren't those found if you just cluster expression data without that knowledge? Because it's washed out by all these downstream effects. Proliferation has been found 1,000 times in cancer papers. Oh, you know, I mean, seriously, I mean, if you look at cancer papers, expression data, they always pick up on essentially cell cycle and how fast the cells are growing. And it's a great indicator of that. It's a downstream effector phenotype. And you have lots of other effector phenotypes that expression data are sensitive to. Stress state of the cells immunological state. All of this stuff is integrated into an expression profile, which is both advantageous or disadvantageous depending on what you want to do. In this case, it was disadvantageous because it's completely obscuring, it's dominating the signal and driving the clustering, whereas the clusters we're finding here are the upstream. You would like to, the hypothesis would be at least think that these are the more upstream causal events that are causing those different subtypes to emerge. But anyway, you can, thanks for the question, because you can find these clusters in expression data in a supervised way. And so if you wanted a diagnostic, for instance, you might now not even use mutations in the clinic. You might use short RNA profiles.
Yeah, probably just to pick up some like eigen features, eigen genes
Exactly.
in your temperature network, and then do the second question is that the last part. So with all the respect, I think that 0.3 is great, and but what's the reason do you think that why you don't get the 0.8 or 0.7 correlation? Is that because of the I know you use decision tree to train the model, or is that because of that, or is because of the initial design of this O gate and the gate stuff?
Yeah, it's a great question. So what I was trying to focus on here is how much better. This is a very hard problem, and it's one that a lot of people have been interested in for 15 years. So I was more resting on the laurels of. I would actually say, if you're interested in this, make sure that that we're right about this. I claim we're right, but don't believe me, because that's a big difference. Okay, that to me, I mean, that'll be interesting, right? If others also are able to build their own hierarchical models and see the same effect. Okay, then the question is, where to go from there? And one is can you learn the functions at the same time? I mean, the beauty of it is we're not learning the structure. The structure is learned. Well, we are, but it's in a separate problem.
Yeah, it's like
so it's like any time in biology, you have structure and function, and we've now cleanly separated the structural reconstruction from the functionalization of that structure. And so my own thinking, by the way, is evolving on this. So rather, in the initial Nexo structure we built for the recapitulation of Go, we did throw in every data set we could get our hands on. Now I'm thinking don't. I'm thinking keep it pure, keep it structural, and then keep all your functional data aside for this to throw at the structure to learn what are those gates on all of those. On that neural network, on that deep neural network of neurons, which aren't neurons, they're components of the cell, but it's the same exact encoding type of encoding.
Yeah, that might work.
So I'll ask a question while we're waiting, which is, how dependent is this on the quality of the data in Go or in any ontology or what sort of.
Yeah,
and
reconstructing this hierarchy of the cell.
Yeah, because it's easy, it's lovely in yeast, it's lovely in cancer. People have been studying it, they grow tissues, phenotype death is really easy to score, blah, 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 blah. There are lots of other much more complex areas about which, we have very little protein data that's a high quality.
Yeah, definitely.
That kind of stuff.
So first of all, just to clarify, it's not dependent on Go at all, because because we threw Go out and we rebuilt it. And then we only bring out, so Go does get used, just to be clear, to make sure everyone's clear on that, Go does get used at the end just as a reference to annotate back and to name the things in our menagerie, basically, right? But
you have to have things in your menagerie.
But then what about the data, so the omics data that go in? What we can do is we can perform so the innovation really was in that clustering algorithm that can build this ontology. At that point, we have standard statistical techniques like bootstrapping that we can apply to assess robustness. So what you can, for instance, do is throw out, so bootstrapping would sort of subsample your data, which in effect throws out about a third of it, and then rebuilds the hierarchy. Does that 10,000 times, and now the terms you want to preserve are the ones that are all, come up in 95% of. So the way it actually works is we build like 10,000 hierarchies and align them all to each other, and then preserve the result.
But how do you know that there's enough data out there to, not the question necessarily of how robust what you built is, but how sensitive or how much you actually recapitulating important interactions that exist, and not.
So through robustness, we're pretty confident that the terms we see are real, and especially when those terms get named because they were in Go. So now you've recapitulated biological knowledge. The more interesting use, though, is now look at the terms that aren't in Go and rank them by robustness. So I think the way you do new biology with this approach is you rank the new terms by their robustness, and you study the top ones first. Now, the other thing you're kind of getting at here is what's missing, and that's a much harder question to get at.
Right. I guess what you're saying is that your robustness, there will be things, there will be areas, tissues, diseases, whatever, where more may be missing, so that when you at the end you look at Go and see what you have, the percentage that are explained will be smaller than others, and that then you'll lean on the robustness analysis.
Yeah, and then you go into lab, right? And at a certain point, you have to go back, and then you really have to start following up on those discoveries. But the nice thing is that it points you at exactly where the novel discoveries in your data are. Or one can imagine in the future is this whole type of, if we can really make this kind of a standard systems approach, then you could be a journal, and someone gives you a big omics data set they've generated just because they can. And you always wonder as reviewers, what's the novelty, and what do people do? They pick an anecdote and they dive deep and follow it up and show you it, yes, fine, it works in a mouse. But that doesn't really tell you anything about systematically how many novel discoveries are in their resource. This does. So I think that's an exciting use of it as well, is it tells you exactly what's novel about the resource.
Great talk. It was really fascinating to
Thanks.
hear all the different approaches. And I had a question about this slide that you still have up here. So the Nexo, it sounded really great, and it was, then when you showed this, I was like, okay, the Go is working out as well, maybe better. And so my question is, what are some of the ways that you think you can add other aspects into the Nexo analysis that would?
Yeah, so actually check out the paper, because we looked a little bit deeper at this in the paper. So now we can dissect. This is blanket for all 3 million genotype-phenotype pairs. But now if we just focus on the genotypes that perturb subsystems in the cell, for instance, DNA repair. In fact, there's a whole experimental piece. We went out and basically did 2,000 more genetic interaction studies to further expand on DNA repair and nuclear lumen, or the two processes. Anyway, as part of that, we showed that for each process, right, for the whole cell, that's the correlation, but for each process, you can get a correlation. And there were some processes where Go wins, and there were some processes where Nexo wins. And if you then start to look at where Nexo wins, it's basically in areas where Go sucks. And so it's interesting. So I think right now, this is not to disparage literature curation. All those Go teams of hundreds of curators, it's not that it's not, you can see it's,
Well, they can't curate
look at that.
this.
Exactly, and they can't curate what has been published, right? Whereas this sort of can, right? And so I think that might be the better take-home message, not to look at the whole cell correlation to answer your question. It's there are some processes where one works way better than the other.
Thank you very much. I'm coming from a different background, not cancer biology, but it seems that the data that you're using for stratification of tumor cancers are somatic mutations. How could you ignore the genetic background of each patient and just including the difference between the tumors and the genetic
Oh, so you mean like the germline or inherited variation
Exactly.
component of each patient? No, so we did ignore it. I mean, by the way, pretty much every cancer sequencing paper that's been published in the past 10 years ignores it. It's a real problem, and I think it's going to be an exciting next 5 years of cancer genomics is to work out the germline. So what can I say? We ignored it, but I mean, we're working from a certain data set that we have in state of the art. Now, the question is, to what degree are these kinds of approaches going to be useful for GWAS types of approaches? And I did show that one example with Joe Gleason's lab on hereditary spastic paraplegia, and that's definitely inherited variants that we're analyzing there. And if you look outside of I mean, this is my talk, my time up here. But of course, I'm, in a way, what I'm talking about here is representative of a field that's trying to do this, in which case, outside of my group, there's a number of examples of network GWAS. But this, as you may know, the verdict is definitely not in on any of that.
So here you're basically comparing each patient with itself, but in other fields, like the field that I'm working on, I'm dealing with the very same problem of relating genotype to phenotype. And there, I'm only using variants that are like germline variants. So basically, there are two types of questions. If we don't want to stratify our patients, and we are only trying to find some genes that are related to that disorder, how should we go from the network approach? How can I narrow down to some genes?
Yeah, so there's a number of, I'd refer you to a number of papers by others. Actually, Eric Shad and I wrote a review article with a number of others a few years ago about all of these kinds of methods. Ed Marcotte has a nice method called boosting GWAS with networks, which is a very simple approach, but a good place to start. I would say if you look at, just to comment about that, so there is a lot of literature on doing exactly what you want to do, which kind of, that motivated us to do something no one had done before, right? But in terms of what's out there, look at the literature. There's a lot of pathway GWAS methods and papers. One thing I would say is most of them are actually not going to be helpful to you. Why? Because they perform the pathway analysis after or downstream of the GWAS. So they've already found p-values of association for every SNP. And then they show that the most significant SNPs are enriched for a Go function. That is probably not going to be helpful to address your question. What your question is can I use pathway knowledge before I compute an association and use that to find genes that I wouldn't have found? And there are fewer papers, but there still are some that do that. So those are the papers you want.
And just last question, the number of genes in your network that you have considered are pretty much less than the number of genes that we have in our genome. So how can we address that question?
Sorry, I missed the question?
The number of genes that you consider in the network, they are pretty much less than what we have in our genome.
But that's by design. I mean, you want
because
the network to be comprehensive.
In 2013 Nature article, you have used only 10% of the edges between the genes, right? In the human net, you have only included 10% of edges, which...
Oh, you're saying we should have used more edges, not fewer edges. Is that what you mean?
Yeah. And those 10% edges only include 7,000 genes, so...
Okay, sorry, I thought your question was why was the network so large? It's why it's so small. Got it. Yeah, that's a reasonable question. So we at the time used the human net database of interactions, and it was very much tied to our analysis of that and what was considered a confident. We were essentially basing our calls on a recommendation of that paper. There might have been a computational efficiency consideration going on as well, but I think it really had to do with what was recommended by that paper. These days there's lots of networks one could use. It's an open question what network one should use. and how exactly to formulate that. Here, you know, rather than pause there, although I think, you know, certainly people are writing review articles about networks and network quality, and my lab is, but here we've sort of raced past that and said we actually don't think networks are the right way to represent the knowledge anyway. We think it's hierarchies. Let's not dwell on it. Let's just keep going, and so that's where we're at.